Hello everyone, my name is Yi. I'm from University of Delaware. Today I'm going to talk about our work of quantifying actin filaments in microscopic images using keypoint detection techniques and a fast matching algorithm. And our work belongs to a session of biomedical and biological image analysis. First, I want to talk about some background of our work. What is actin filaments? Actin filaments is a family of proteins that form microfilaments which performs many conserved functions within a cell. It is found in essentially all eukaryotic cells. Actin participates in many important cellular processes, including muscle contraction, cell motility, cell division, and the establishment and maintenance of cell junctions and cell shape. As you can see from the image on the right, the purple ones are the actin filaments. It shows how the actin filaments interact with other objects like microtubule or stromules. So knowing the matrix of actin network is very important to studying the actin dynamics and their interactions with other cell activities. And to achieve this goal, it requires approaches can successfully to extract individual filaments. So the domain expert could quantify the length, curvatures, and number of active filaments and know how they will change under different influences, such as different chemical influence, how their number will change along the time, and so on. However, there are some challenges for this task. First, the macroscopic images are usually with high level of noise, and including some optical blurring, clutter, and overexposure areas. And secondly, actin filaments are very dense and complex. Individual filaments share similar features, and this makes it extremely difficult to segment them at an instance level compared to other common objects like cars, person, and etc. Therefore, popular anchor-based instance segmentation method like MOSC-RCN is not suitable for this task. Figure 3 shows that example why the anchor-based instance segmentation approach were not suitable for the filamentous structures. Because anchor-based approach, they require a bounding box, and in many situations here, the bounding box were overlapping with each other for different active individuals. Like this purple and green one, they are different filaments, but they share exactly the same bounding box. And there's no way the mask RCN can separate them. There has been some works working on this problem, and most of these works are non-deep learning based approach. We summarize this approach into a pipeline, which including 3 steps. So most of these works, they first use traditional image process techniques such as canny edge detector, linear Gaussian filter, and contour-based approaches to obtain a binary image segmentation first. And then they scalarize the binary segmentation and find the intersection points. And the steps 3, they break those intersection points and run disconnected components to obtain individual segments. However, there are some problems of this approach. First is that those approaches expect a very high computational burden. And second is that the segmentation result highly relies on the hyperparameter adjustment and can be highly influenced by the noise pixels. Third is that disconnect acting at junctions of skeletonization creates many small and short segments which are incorrect and can highly impact the future quantification analysis. As you can see from the figure 4 here, after skeletonization, original intersections here, one intersection turns into 1, 2, 3 intersections, and this one turns into 1 and 2 intersections, and the number of segments increases a lot by just disconnect at intersections. There are some deep learning based approaches, But most of these deep learning-based approaches only focus on binary segmentation, not instance segmentation. And those deep learning-based approaches can achieve a very good result on binary segmentations. Our motivation is that if deep learning-based segmentation outperform the result of traditional approaches, can we use deep learning-based approach to do instance segmentation? And also, can we utilize the binary segmentation from the deep learning-based approach and work on that further to obtain the instance segmentation? Second is that if we want to use deep learning-based approach, how can we obtain the instance segmentation without any instance level labels? Because it requires a lot of work to label the actin filaments at instant level, and because each image contains hundreds and thousands of instances. Inspired by the traditional approach pipeline, we were thinking, what if we can successfully detect the intersections? Can we avoid the problem of scatterization, which caused many false small segmentations? How does that help us to obtain individual filaments if we can obtain the exact intersections? Our observation is that real intersections is not just one point, but an area with different shapes, like right here. Some intersections are large, some intersections smaller. It's not just a point, but it's an area. So this probably is the reason why scatterization can fail to capture the exact intersection areas. So another observation is that as long as we can successfully detect the center of the intersections in the mask, we can use this center as our origin and march out along the segmentation mask. Inspired by the human keypoint detection techniques, we can just use a convolutional neural network to predict the intersection points. We can use each intersections as an origin of the wave and march along the mask. Two waves from different origin will meet at the center of individual segments, and the travel distance of the center will be half of the value of the true length. So utilize the intersections and the binary segmentations, we can obtain the individual segments. Therefore, we will have our proposed pipeline here. The original image will be fed into a neural network to obtain the binary mask, and the binary mask will feed into a keypoint detection neural network. And this will output heat map and the offset map. The offset map helps the heat map to pin down where the exact key point center is. And then once we have those intersection points, we can use this intersection point as the origin and use a fast march algorithm to obtain a distance map. As you can see from the image F here, the middle of segments will be the value of half of the total length. And thus, we can know how many segments are there and without disconnect them. As you can see from the figure G here, the number of local maxima will be the total number of segments, and the value of each local maxima will be half of the length of the segments. Now we're going to go through the details of our approach. For the binary segmentation, we directly adopt the network and pre-trained weights in the paper density connected stacked unit. For the keypoint detections, we use ResNet as the backbone, and we adopt the strategy in PersonLab. So the output will be a heat map of a disk around the intersection points, and an offset map, which is the offset value around the intersection point. So the pixels around the intersection point will be pointed towards the intersection. So by half vaulting, we can refine the exact location of the intersection. However, there's no existing dataset for intersection detections in filamentous structures. So we created a synthetic dataset for intersection detection. We created 10,000 images with size of 256 by 256. Each image randomly included 1 to 6 filaments, and we intentionally randomly generate X junctions and T junctions to make our synthetic dataset more realistic, and the shape of the curves are randomly generated. Once we have the synthetic dataset, we can train our network, and then we can run the quantification analysis using a fast marching algorithm. We set each intersection point as origin and then march out along the masks. The figure on the left is the distance map. Take this as an example. The wave will march out from the origin and reach the highest at the center of the segments. And the image on the right shows the highest value when two waves meet at the center. And this value will be the half of the length of this segment. And by counting the number of highest value, then we can obtain the number of segments. Now we are going to present the analysis and the results of our approach. Because there's no ground truth for our dataset, we can only use the percentage difference of length and count as a metric to compare our approach and others and see the effectiveness of our approach. As you can see from the table 1, the difference of total length is relatively low because binary segmentation results are similar, and the critical difference depends on the number of filaments. So X works well for long-acting filaments, and the author will group different contours to form a single long-acting filament. But actin filaments in our microscopic image are very short and dense, and each small segment is considered as a single actin filament. So X has the lowest count per image and has the maximum difference number, so the percentage difference of count is very different from our approach. We also manually count the number of filaments for two images to show the effectiveness of our approach, one with the high percentage difference and one with a low percentage of difference of the count. As you can see from table 2, our proposed approach shows a closer count result to manually count. Figure 5 here presents a qualitative result of junction detection. Our proposed approach detects junctions more precisely, and our approach can avoid counting small false segments caused by scatterization. The left image here is our approach, and the right image is a traditional scatterization approach. Those thick dots here are actually contains multiple intersections. Our approach is much more precise than traditional scatterization approach. Conclusions. We have shown that our approach can successfully detect junctions and endpoints of actin filaments network, which is fundamental for estimating length using a fast marching algorithm. And we combine deep learning models and a fast marching algorithm to automate estimating the number and length of the actin filaments in microscopic images. We proved that our approach is practical by comparing against the other approaches. Our approach is relatively faster compared to other approach and requires less parameter tuning. With individual filaments obtained, we are able to quantify other metrics like curvature and average intensities. We hope that our work can inspire other researchers who work on similar problems, even though the settings may be different for different domains. Thank you so much for your listening, and please feel free to email me for any questions, and welcome to join the discussion session later. Thank you.